All right, it's 11:45. You know what that means. That means I got to make you do some problems today. So we're going to work through some of these exercises. But first, a few quick announcements. Let's see. Is anyone still counting their F1 flies? A few people, maybe? Ooh, we are so close to 1,000. If we just get a few more, that would be really nice. Just to satisfy those big number urges. So anyway, F1s are almost done. You will probably be counting F2s starting into this week or early next week. Either way works. Count as many F2s as you can. So 1,000 won't be quite enough for this experiment. So you're just going to have to count. Let's see. Also, so that's what this week is in the lab, go count flies. Also, this is kind of rushed, and the Thursday labs already know about it. But I rushed to get another cross going because I realized we were converging on a collision course with spring break. So the other thing you'll be doing in the Tuesday lab is setting up a cross of white miniature forked by wild-type flies. Thursday lab has already got it done. It didn't take long, right? Who's in the Thursday lab? Piece of cake, right? You're so used to this. You know how to make medium. You know how to set up a cross. So yeah, we're just going to do that this week. It'll take 10 minutes. That'll be easy. All right. Then the other important thing this week is there is an exam. I will give it out on Wednesday. It'll be due on Friday. So another take home. It's going to cover everything we've covered in the course so far. You don't ever get to forget Mendelian genetics. Sorry. You gotta keep it in your head all the way through the rest of the semester, otherwise nothing makes sense. So we'll be doing that. I gave you a little preview of some of the kind of material that will be on the exam and the homework problems for this weekend. And what I want to do today is go over those. Make sure everyone's 100% clear on how to do them. And I've also thrown in a few other simple sex linkage problems for you to think about. So that's our agenda for today, which means I will present the problem to you, and I'm going to make you do all the hard work. You're going to have to solve these for me. For the first few, it'll be easy because you already did it with the homework, right? So everybody's 100% ready to go on this. So let's take these little homework problems apart and see how they go. Oh, also, I was asked earlier, notice it says, turn it on. It says chapter 5 for Wednesday. Yeah, that will not be on the exam. So you don't have that. We're about to get into linkage, and that's going to take a while to work our way through and grasp the concepts. So no, that will not be on the exam. It's all going to be monohybrid, dihybrid, trihybrid, tetrahybrid crosses. It's all going to be stuff like sex linkage, other modes of inheritance besides dominant and recessive. All those things will be on the exam. Also, there will probably be a chi-square problem on the exam. Aren't you relieved I didn't hit you with chi-square on this weekend's homework? I figured you needed a break from it. But yeah, we're always going to be throwing chi-square problems at you. All right. So, here is one of our problems. So I told you, okay, we got vestigial. That's a mutation. It's on chromosome 2. When I tell you that, that means it is not a sex chromosome thing. So it's not on the X chromosome, it's not on the Y chromosome. That would be chromosome 1 in flies. So I tell you chromosome 2, you immediately know, oh, that's autosomal. And I tell you, okay, white is a gene on the X chromosome, which tells you right away, okay, this is going to be a sex linkage problem. And then I set up the problem. You have a white-eyed bale with long wings. that you crossed to a red-eyed female, also with long wings. Okay? And then you start hatching out lovely little babies, and you discover the first few progeny. Some have short vestigial wings and some have white eyes. Haven't done anything quantitative yet. You just see there's a few vestigial winged F1s emerging, a few white-eyed F1s emerging. So now you should be able to figure out the genotype of the parents. Right? So what's the genotype of the female? And how did you arrive at it? Yes?
Heterozygous for both of them?
Heterozygous for both. Yes, so the female you're saying is heterozygous for vestigial, so it's VG plus, VG minus, and also it is heterozygous for white eyes, so it's W plus to use that abbreviation, or XW plus, XW minus, whatever. Okay, were you correct? Why, yes, you were. Pretty easy, right? So this is a non-quantitative problem. You just need to know what kinds of babies you get out of this, and that tells you right away. Okay, they've got to be heterozygous. Why? Why do they have to be heterozygous?
Because the first few progenies have short wings, so they have to be heterozygous for.
Yeah, so you have a few of the progeny that are expressing the recessive phenotype. And so the only way they could do that is if they got a combination that made them homozygous for that recessive phenotype. Okay, what about the male? What is the genotype of the male? So the male, again, is long wings and white eyes.
XWY and then heterozygous for wings.
Sounds good to me. Yep, so that's what we got. So now we got the information we need to do the rest of the problem. So here's our next problem. So we have those flies go in it for a while, they produce 1,000 progeny, just like the F1s in your lab right now. Anyway, you get 1,000 progeny, and we're going to ask how many of each of the following phenotypes would you expect to see? And just to remind you, there's our genotypes. Now, I got a few questions by email from a few of you about the homework, and that's fine. Do feel free to email me with a question if you're struggling with something on it. And there wasn't much I could tell you. Yeah, you have to apply the principles we've been talking about all semester long, that you can just take these and treat them independently. Okay, so we got, if we just look at X, for instance, there's an X heterozygote, there's an X hemizygote, it's got white and Y. Just take that, solve that problem first. And then we got a heterozygote for vestigial, another one here, heterozygote for vestigial. So let me ask you, what frequency do you expect of vestigial versus non-vestigial?
75% will have long wings and 25% will have vestigial.
Exactly. So this, the vestigial part of the problem, is just this boring, simple monohybrid cross, isn't it? You know how to do that. The white-eyed cross, a little more complicated because we have to take into account the sexes. But this should tell you right away, all I need to do is apply that fork line method. So I think, yeah, there I got a diagram for you. So we're going to get half males and half females, right? Of the males, let's look at them up here. So first I'm doing the red and white eyes. So we got this female crossed with this male. We're going to get 50% red-eyed, 50% white-eyed. When we look at the females over here, that's going to produce 50% red and 50% white. And then we just had explained to us we're going to get a 3-to-1 ratio for vestigial, so there it is. So you can just multiply those all out. And that will tell you the frequency of each of the different phenotypes. Okay. So who did this for red long-winged bales? Go ahead.
187.5.
Why, that sounds exactly right. Yeah. So it's just 0.5 times 0.5 times 0.75. And we get that nice result. Let's skip ahead to, the red vestigial males.
It's the same for the females.
Okay, guys, go through all of these. No, it's not the same. With the red vestigial males, it's going to be 62.5. So again, look over here. Long-winged, it's going to be 75% of the total. And the vestigial winged is going to be 25%. So if we go take 0.5 times 0.5 times 0.25 times 1,000, and we get 62.5. Okay, the rest of it is just, it's boring, right? You guys know you learned how to do this in 3rd grade or something, right? Multiply 3 numbers together. So that's all we do for each of these. And we get that set of numbers. So that's what I was looking for in your answers. Pretty straightforward, I hope. Again, the magic formula is, whenever you get a complicated problem, break it down into 2 or more simpler problems and multiply the results together. So on the exam, I could give you some horrendous thing where I've got white eyes, which is sex-linked. I got another trait which is got incomplete dominance, and I throw in another one just for the heck of it. Could get a whole set of these. It looks terrible when you sit there and just look at the problems, but you now know the secret. You just split it apart. Take each part a piece as it is, and each individual part is really, really simple. Okay. I gave you some pedigrees. Oh, this will be interesting. So let's look at this first one. Right there. And ask, what is the most likely mode of inheritance of the trait? And there's a bunch of possibilities I gave you. What did you say? Okay.
X-linked dominant.
X-linked dominant. Why did you think that?
Because in order for the parental generation to have infected female children, if the male is infected, that he only has the X to donate, and so that's where it has to be, because he can only give that to make female children.
Right, so if it's X-linked dominant, all of his daughters will express that trait. What about the boys? How come the boys don't have it?
They inherit the Y.
And there's mom, she's wild type. She's got the ordinary allele, whatever that may be. What about down here? What's going on here? So here you got a son of this group who presumably is not got it then, right? Because it's X-linked dominant. Here's a woman. He finds a woman who's got the trait. And how do you get that distribution? Yes?
A mom can pass it to both her daughters and sons. Sort of half-ish distribution.
Yeah, so again, humans, we don't have enough babies to really tell. But here you see, we've got a roughly half and half. It's roughly equally distributed between males and females. So that's another clue. So what does that tell us? So it's X-linked dominant. All right, all the affected man's daughters are affected, none of his sons. And the affected woman here passes it on to half her sons and daughters. That tells you right there, okay, this has got to be X-linked dominant trait. Okay. Here's another pedigree. What's this one? Anyone? Somebody who hasn't answered already. Be brave.
X-linked recessive.
X-linked recessive. That sounds like a good answer. Why?
Because if the grandma has
two of the recessive alleles, she has to pass it on to all of her sons, but the females won't get it because they don't have two of those.
Wait, she has to pass it on to all of her sons?
Yeah, because they'll get.
Oh, because they'll get the Y from dad, so they have nothing to oppose that allele from their mother. So all the sons have it. What about the daughters?
The grandpa doesn't have it, so
Uh-huh.
he doesn't have it on his X, so he can't give it to have both have the allele.
So all their daughters will be wild type phenotype. What will they be genetically though? They're going to have to be homozygous for that trait, right? So we're going to get, so this one is homozygous for that trait. She's passing along one X chromosome with the mutation. The father is passing along one X chromosome without the mutation. So each of the daughters has to be a carrier. Okay, is that confirmed in the third generation down here? So let's look at this one over here. So this woman, we're saying, is a carrier. This man does not have it at all, maybe. And when you look at the children, you see, oh, she passed it on to her son. All the others don't have it. Okay, the daughters down here, are they carriers or are they free of the trait?
Depends on the X chromosome they got from the mother.
Yes, there's a 50-50 chance. Right. And then this is another interesting test case over here. Here's this gentleman, he's got it. He finds a wife somewhere out there. There is no double line here, so it's probably not consanguineous. And of course, then all of their children also have it. In many pedigrees, when you see a pedigree like this, you'll generally see that when they marry outside the family, they don't express it, which means it's a rare trait. If it's something like this, where we've got individuals who are finding mates outside the family, and they've got it, it may mean that this is a very common sort of trait. Okay. Did I answer it correctly? Yes, I said it was X-linked recessive. And that's because it can skip generations, and it's X-linked because sons are more frequently affected than daughters. Okay, another part of the question though was to identify the genotype. So what are the genotypes of this individual, for instance?
Homozygous for the trait?
Homozygous for the trait, yes. What was the other one I mentioned? I said this one. Number 11. Go ahead.
XY.
Yeah.
Yeah.
So he's got a wild type X and a Y chromosome. Let's just, I don't remember which ones I asked you, so I'll just shoot ahead here. Yeah, there we go. So you can see this individual was a carrier. This individual was homozygous for it. Here's a male that's got the trait and a Y chromosome, etc. So that's another kind of question I can throw at you on the exams. Here's a pedigree. Tell me what all the genotypes are. Probably not all of them, because that would be tedious to grade, but you get the idea. Okay. So everybody got an A on the homework, I presume. You seem to know what you're doing. What about this one? Here's another problem. That wasn't on the homework. All right, so I set this up and tell you, you've got an insect species, a mysterious insect species. We don't know which sex is heterogametic. Hey, if you were Nettie Stevens, how would you answer that? How would you figure out which sex is heterogametic?
Test across?
All I'm saying, if you were Nettie Stevens, remember she was a cytologist. She would just find a male or a female and she'd squish their cells and look in the chromosomes and see which one had the mismatch. So that's another approach to take. But yes, we could do a test cross and see what we get. Well, except just to determine sex, it's always a test cross, right? The homogametic sex is always basically recessive for two loci, and the heterogametic sex has got a, anyway, we'll see, we'll work through it. Okay, so we got X-linked recessive mutation, which causes reduced wing. We are now going to ask, okay, using just purely genetics, we're not going to cheat and do the Nettie Stevens things of squishing cells and counting chromosomes. We're going to do this with pure genetics. So contrast the F1 and F2 generations between a female with reduced and a male with normal-sized wings. So this one's a little tricky, you got to think it through. So we're going to make a supposition here. What if the female is the heterogametic sex? What would the answer to this question then be? Well, we can figure out the genotypes of the female. There's our female. She must be, the female must be heterozygous because we've just said, okay, we got a reduced wing, we got a female with reduced wing, so she must look like this because she's heterogametic, right? And the male, because it's wild type in phenotype, it must be this. Again, this is the homogametic sex. Another important note here is, if you see a problem like this, just assume they're homozygous, unless they tell you otherwise, or give you a prior genotype from the parents so you can figure it out. Okay, so that's one thing we figured out. That must be the genotype of these individuals. But now we're doing the test. And we ask, what is going to come out of this? Okay, what are the F1s going to look like? Here they are. All the males will be X plus XRW. So remember, males are the homogametic in this example. So we're going to take one X plus, and we're going to take one XRW from the female here. So all the males are going to look like that. So they're going to have normal wings, but they're all going to be carriers. The females, on the other hand, there's one right there, gets a Y from mom, and the X plus comes from dad. So they're going to have normal wings. Is that all making sense so far? Okay. So tell me, what are the F2s going to look like? So the F2s are going to cross this by this. Yes?
Half the females are going to have normal wings, and the other half's going to have reduced wing.
Okay, of the females, so we're starting with the females. All right, so in the F2s, they're going to all inherit X plus from mom, right?
Oh, I thought they were going to inherit Y.
Oh, you're right. They're all going to inherit the Y. I got that backwards. You got it right. And so half will be X plus Y, half will be XRWY. So they'll all be, well, half will be reduced wing and half will be normal wing. Okay, what about the males? Well, I already gave it away. So the males are going to inherit that X plus, so they're all going to have normal wings. But half of them will be homozygous for the normal wings, half of them will be carriers for RW. Did y'all get that on? Yeah, get that in your notes? Okay. I will be posting this whole file on Canvas right after class, so if you miss something, it'll be there. Okay, but now we change our assumption. What if the male is the heterogametic sex? What will be the genotypes of the parental generation? So again, we got the same phenotypes up here. So we got a female with reduced wings and a male with normal-sized wings. And now in this case, this is the heterogametic sex. So what will their genotypes be? Go ahead.
Male will be X plus Y, and then the female will be homozygous for reduced.
Yeah, very good. Okay. So yeah, we get this genotype. And again, a hint for this kind of problem, it helps to be really methodical and first figure out what the genotypes of the parents must be. And then everything else just falls out easily. Okay, what will the F1s look like from this cross? Yes?
All of the females will be heterozygous, and then the males will be the reduced wing Y.
Right, so all the females will get one of these reduced wing X's, and they will also get an X plus, so they'll express the wild type phenotype, but they'll be carriers. And what about the males?
They'll be reduced wing and Y.
And the males will all inherit this X, RW, and a Y, so they'll all be reduced wings, 100%. Okay, there's our F1s. Did I get it right? Yes, I got it right. Okay. I'm glad you guys are double checking my work. It helps. All right. F2s. What do the F2s look like? So we're going to cross this by this. Yes?
Half the females will be carriers and half will express reduced wings.
So half will be, okay, we're talking about the females here. So the females will all inherit this. And half of them will get this one, and the other half will get that one. So yes, we'll get a split. All right, what about the males? They're all going to inherit this Y. And?
Half will have normal wings and half will have reduced.
Exactly. Okay. It all worked. Man, these problems are so easy. You guys are going to sail through the exam, and there goes my grades. They all get inflated. It's okay. Okay, here's one. We mentioned Calico Cats before. Now remember, Calico Cats were the product of dosage compensation, right? So you got either a black or an orange-yellow pigment gene, an allele, on the X chromosome, and one X chromosome gets randomly inactivated. So here's our problem. We got a Calico Cat, and this Calico Cat has been sneaking around, and we don't know what the father cat looks like. But we get these kittens out of it. So we get one yellow male, two black males, two yellow females, and three calico females. And we're going to ask, what is the color of the father? So we can hunt him down and get him neutered down at the vet's office, right?
Yeah.
Yeah. These irresponsible pet owners. Okay. So what color is the father of the litter? And how do you figure that out? I'll give you some hints. So here's the first hint. Just ignore the male kittens, okay? So they inherited a Y chromosome from dad. And Y chromosome doesn't transmit pigments. So the male kittens, they're going to tell us absolutely nothing about what kind of X chromosome the dad has. So this one, just scratch it off. That one, just scratch it off. Okay, what about these calico females, are they at all useful in figuring this out? How do you get a calico? You get a female cat with 2 X chromosomes, right? One from mom and one from dad. And then they randomly inactivate one of the 2. Does that help you here? No, because you don't know whether it was dad's X chromosome or mom's X chromosome that got inactivated. So we can just ignore the calico kittens too. So we scratch those off. Which leaves just the 2 yellow females. So what does that tell you?
Male has to be yellow too.
Yeah, so these kittens have 2 X chromosomes, and on each one they got a yellow allele. So it doesn't matter which one they inactivate. They'll still be yellow. So yeah. So all we need to do is look at those yellow kittens, and we see that you got these yellow kittens because one or the other was knocked out, and therefore dad was a yellow tomcat. And we call up the Stevens County Humane Society, and we tell them, go look for this cat in my neighborhood that's impregnating other cats, and capture them, take them back to the vet, get them cleaned up, give him his vaccines, and neuter him. Okay. Yes, a little lesson in Humane Society practices here. You do know we have a Humane Society here in town, right? Yes, you can call them and talk to them anytime. Okay. Here's a related problem. So in this case, we're going to use a little technology, and we clone a cat. This cat got the name Carbon Copy. This is a real cat. This actually happened. And she was cloned from a Calico Cat. And when she was born, she had nothing but black patches and white patches. And remember, the white patches are all independently specified. But she didn't have any orange patches at all. So she didn't look like a Calico Cat at all. She looked like a black cat with a white chest. How does the lion hypothesis explain this result? Does everybody remember what the lion hypothesis is? Nobody is shaking their head yes. You've forgotten what the lion hypothesis is. This idea from Mary Lyon to explain X chromosome inactivation. So here, I'll give you a quick reminder. So there's the lion hypothesis. So you got 2 X chromosomes in a female. The lion hypothesis proposes that which one gets inactivated is random. Just flip a coin, see which one gets inactivated. And the choice is made early in development. And once the choice is made, all the progeny of that cell have the same X chromosome inactivated. So now, can you explain this result, how a Calico Cat, when cloned, produced a black cat? What does cloning a cat entail?
It's got the same genotype as...
It's got the same genotype. Where do they get the cell that you clone?
stem cell?
Yeah, but from an adult, right? Mm-hmm. So in this case, we cloned a daughter from a single, I think it was an epithelial cell, so a skin cell from the adult. Look at that second point there. So what would you infer from this? That epithelial cell had already made the choice, right? It was already committed. It already shut off the yellow chromosome and was only making black pigment. And then also, this third point is also relevant because once the cell made that commitment, all of its daughter cells also had the same chromosome inactivated. So, that's our explanation. The clone cat was made from an adult cell, in which the choice had already been made, which its chromosome would inactivate, and all of its progeny would have retained that choice. There's a secondary question there. Could the clone cat have turned out orange? Yeah, it's just random which one you got the cell from. Here's a more advanced question. Could this be a bad thing? Could there be consequences to the cloned cat produced by this method? What could they be? It's genetically identical.
There could be deleterious mutations on the chromosome that gets activated.
Right. Yeah, so often in normal individuals, so like all you women out there, you are a mosaic, a patchwork of cells where some have turned off one X chromosome, some have turned off the other one. So if there's a deleterious mutation on one X chromosome, still half your cells have the non-deleterious allele. So they'll be fine. So there's a possibility this would not be entirely desirable unless you could figure out a way to reset that X inactivation. Okay. Man, you guys are just acing this so far. All right, here's another one. So now let's talk about people. We've mentioned there's one trait, color blindness, that's carried on the X chromosome, and it's recessive. So here's something sneaky. We say a man whose father was colorblind has a colorblind daughter. You know, this is the kind of question we're asking. Well, why didn't you test the father? Figure out directly whether he's colorblind. This sometimes happens, though, if, for instance, the father died before you were doing the testing. Okay, the question is the man... Another thing here, this entire question is centered on the father. What about the daughter? But anyway, okay, so we're going to be centering all these questions on this guy, this man, whose father was colorblind. Is he colorblind? Yeah? How do you know?
The daughter has to get 2 copies of the recessive.
Right. Yeah, so somehow the daughter had to have gotten 2 colorblind X chromosomes, and one of them came from her dad, so we can assume that. Just to let you look ahead, we will later talk about some other sneaky mechanisms where that doesn't necessarily happen. Yes?
Does that mean that the girl's grandmother was a carrier for color blindness?
Oh, you're jumping ahead to this question. Let's do that. So first of all, yes, so probably the father was colorblind for the reasons we just gave. And then the next question is, where did dad get his gene for colorblindness? Because right here we said a little red herring, isn't it? We said his father was colorblind. Is that relevant? No, because he's not going to get an X chromosome from dad, from grandpa. So that was devious, throwing that in there. But you rightly ignored it. Very good. So where did he get his gene for colorblindness? You came up with the answer.
His grandmother.
Yes, his the daughter's grandmother, his mother, must have given him that trait. Okay, so most likely from his mother, who had to have been a carrier, not from his father, would not have passed an X chromosome to his son. All right, must the fathers of all colorblind girls be colorblind, and why? Don't be shy.
Unless a weird mutation happens, yes.
Okay, yeah, there is the possibility of mutation. So good thinking outside the box here. But in general, yes, usually And like you just said, not always. There's ways we could expect a mutation to spontaneously arise. But in general, colorblind daughters had to inherit a colorblind X from mom and a colorblind X from dad. Again, you notice all the answers to these, I read this problem, I said, probably, most likely, and usually, because there are occasions where there's funny, funky things going on, and we'll talk about those later. Fortunately, they will not be on the exam either. That's all you care about, right? Don't worry, I'll get you later. Okay. So here's another question about an excellent trait. So hemophilia is inherited exactly like colorblindness. That is, it's an allele on the X chromosome. The dominant allele calls for normal clotting time of the blood, and the recessive causes hemophilia. So in hemophilia, blood clots more slowly or not at all. So it's got all kinds of problems. Now you are a genetic counselor. Great promotion. Oh, you should be getting paid more. Anyway, so we got a normal woman. She marries a normal man. They have 1 daughter and 14 sons, all normal. Okay, this is carrying the principle of doing extensive genetic testing a little far. So she had 15 kids. In view of this, so all of her kids are normal. What gene combination would this woman most likely have? Is she a carrier for hemophilia, you think?
She could, but we wouldn't see it in their offspring if the man doesn't have it and she's heterozygous.
Oh, but remember, the man passes on the Y chromosome.
So probably not.
Yeah, if she was a carrier, you'd expect half of her children, half of her boys, to be hemophiliac. Now look at that, 14 boys, wow. And they're all normal, so that makes it statistically highly unlikely that she's a carrier. So she's free of the disease. And of course, on an exam, I could have said, okay, do a chi-square test to show me how likely this was. Okay, in view of this, what gene combination does one most likely have? She's almost certainly wild type on both X chromosomes. What is the probable genotype of their 1 daughter?
Homozygous for the dominant allele.
Yeah, she's probably got 2 copies of the wild type allele, one from dad, one from mom. And she genetically, on this one locus, she would look just like her mother. Okay, that's nice to know. So, if you were a genetic counselor and this woman came in and asked, okay, what's going to happen with my 16th child? You would say, no, you don't have to worry, they're probably not hemophilia. I'd say there's other things to worry about with 16 kids. Okay. Yeah, so she's homozygous for the wild type allele. She's thoroughly tested this cross. So we can predict that the mother and the daughter both look like this. Okay, and I'll remind you, if I ask this kind of question on the exam, I probably have a little block where I'm asking you to explain how you arrived at your answer. And these are all examples of how to answer very briefly. You don't have any kind of thing that says it's an essay question on these exams. You don't have to go on for 2 pages, right? This is good. It'll save me from going blind as well. Okay, here's a similar sort of question. So we got a young lady, she comes to you for advice. You're a marriage counselor now. And she tells you that her brother has hemophilia. I'm sorry to hear that. But both her parents are normal. She wishes to marry a man who has no history of hemophilia in his family. And she wants to know from you, what is the probability of her children having this disease? So what would you tell her? You'd sit her down in your office and carefully explain this. Maybe have some diagrams, all this kind of thing. So what would you tell her about the probability of having a hemophiliac child from this marriage? So again, all we know is she's got a brother who's got hemophilia. Yes?
Would she herself have a 50% chance of being a carrier?
Yes, so there's a 50% chance she is a carrier. But that means there's a 50% chance she doesn't have the hemophilia allele at all, right? If that is the case, then you would just pat her on the hand and say, don't worry, you're not going to have any hemophiliac children, right? Unfortunately, there's the tests for this aren't really widely accessible. So if you suspect you're a carrier for hemophilia, there are things you can do. There is a blood test that you can take. It's not 100% reliable, it's more like 80% reliable. If you're a heterozygote for hemophilia, your blood clots a little slower than if you're a homozygote. So you can do that. If you got money to spare, you can go out and get your X chromosome sequenced and look for characteristic alleles there. Those don't work. But anyway, if she doesn't have the allele, clean bell health, you're not going to have any hemophiliac children. What if she's a carrier, though? Yeah.
If she's a carrier and the man she's marrying doesn't have it, her daughters would all be fine, although some could be carriers.
Right, 50% would be carriers.
But then 50% of her sons have the possibility of expressing it.
Yes. So you got to break some bad news to her there. So as long as she marries someone who's not a hemophiliac, the man is only going to contribute a wild type X chromosome, so that can cover up some problems. Second, all that matters is whether she is a carrier or not. As I said, there's tests that you can take to try and evaluate that. So there's a few things out there. I usually don't hear about them being used much. Okay, if she is not a carrier, there's a 50% chance, and there's essentially a 0% chance that any of her children would inherit it. If she is a carrier, there's a 50% chance of that. Then there's a 50% chance that any son will be hemophiliac, and a 50% chance that any daughter would be a carrier. So that means that without knowing her genotype, as she's sitting there in your office, you can tell her that right now there's about a 25% chance that any son would have the hemophilia disease. So, which means there's a 12.5% chance that your next child would have hemophilia. And if you're like that previous woman, and you have 14 sons, pretty good chance you're going to get a hemophilia out of that, right? Okay, so this is another thing to remember. If you don't know with absolute certainty what the genotype of the parent is, and you can just say there's a 50% chance that they're a carrier, then I ask you, what is the chance of her children having? You have to multiply those together as well. Okay. So that's our answer there. I think that was, yes, that was it. We finished early because you guys just raced through these. You know what you're doing. Are there any other questions that you might want to be prepared for our exam? As I said, it's going to cover everything in the class to date. You guys know monohybrid crosses, right? But there could be dihybrid, could be trihybrid, could be combinations. And again, the magic secret is just break them down into individual pieces, and it all works out. Okay, I will see you all on Wednesday then.
Oh, I
I was like,